I don't know what to say. I can start off by saying thank you to Ed and to Bill and to Al Merrill for nominating me and for the awards committee to bestow this honor upon me. So we're here to celebrate the legacy of Herb Tabor. Herb was a JBC author for 72 years. He published his first paper in 1943, and he published his last paper in 2050. He was known for his seminal studies on the biosynthesis of polyamines and their properties in controlling the growth of normal and cancer cells. He dedicated his life to the JBC. He became an editorial board member in 1961. He became an associate editor in 1968. And from 1971 to 2010, he was the editor-in-chief. He was the co-editor from 2010 to 2020 with Marty Fedora and Lila Gavrish until his passing in 2020. I thank Herb for supporting me as an author, as a reviewer, as an associate editor. And those of you who know him, I think you'll agree with me that he was a great man. I'd like to thank my formal and informal mentors, Bob Levin, who was my PhD advisor, and Bill Dohan, who was my postdoc advisor, and then Robert Bell, Susan Henry, and Ed Dennis, who were my unofficial mentors. Together, they helped shape my career and my life. I wouldn't be here today if it weren't for the postdoctoral fellows and my graduate students and undergraduate students who worked so hard, and it was an honor to be their mentor. I had a lot of collaborators during my career, and their names are there. And I want to especially acknowledge Susan Henry, who worked with me on the regulation of phospholipid synthesis, Dennis Volker, who worked on the molecular characterization of many of the phospholipid biosynthetic genes that Bill talked about, and most recently Simeon Sinisioglu, who worked on the NEM1-SPO7 PA phosphatase complex that I'll talk about today. My research has been funded by the National Institutes of General Medical Sciences, and I'd like to acknowledge Jean Chin and Zhongxing Nie for supporting my merit and my Ramir Awards. So let's begin with the science. So I started my career in lipid metabolism when I joined Bill's lab at the UT Medical School in Houston. And at that time, the fluid mosaic model was a fresh concept. And Bill's lab was interested in studying the biochemistry and the molecular biology of membrane phospholipid synthesis in E. coli. And he gave me the project of studying the kinetic properties of the E. coli phosphatidylserine synthase, and that was my first JBC paper. In establishing my independent lab at Rutgers University, I segued over to using the model system yeast, Saccharomyces cerevisiae. And I'd like to acknowledge the foundational studies of Susan Henry, Robert Lester, and Satoshi Yamashita, for which was the basis for my early studies. I also show you the photograph of Eugene Kennedy at the bottom left of the slide. Eugene Kennedy was responsible for identifying many of the reactions in the pathway. You should know that Eugene Kennedy was the postdoctoral advisor of Bill Dohan. So Gene Kennedy happens to be my grandpa. So Bill talked about some of the things that we did in yeast, but I want to focus in on some of the collaborative efforts with Susan Henry in looking at the regulation of the phospholipid biosynthetic pathway. And our studies led to the observation that phosphatidic acid, or I have PA or phosphatidate, turned out to be a very key regulatory hub in the regulation. And so with the encouragement of Bob Bell, we began our studies on looking at the enzyme that controls the metabolism of PA, and that's PA phosphatase. So PA phosphatase catalyzes the dephosphorylation of PA to form diacylglycerol. In the seminal paper by Kennedy's group in 1957, he showed that the diacylglycerol could be used for the biosynthesis of triacylglycerol, and it could also be used for the biosynthesis of phosphatidylcholine and phosphatidylethanolamine by the CDP-choline and CDP-ethanolamine-based pathways that Kennedy discovered. Well, I know you want to know what George Carman was doing in 1957. So I was in the second grade at public school number 18 in Jersey City, New Jersey. So over the years, our laboratory had discovered 4 genes encoding for PAP enzymes. And 2 of the genes encoded for magnesium-dependent PAP enzymes that catalyze their reactions based upon a haloacid dehalogenase or HAD-like domain containing the canonical DX-DXT motif. And then 2 of the genes encoded for magnesium-independent PAP enzymes that catalyze their reactions by a 3-domain lipid phosphatase motif. And by the way, that was discovered by a postdoc fellow in my lab named Joe Stuckey. So remember that name. So what is the contribution of each of these PAP genes to PA phosphatase activity and also to lipid metabolism? And through the characterization of various mutants alone and in combination, Gil-Soo Han and Ming-Jong Che showed, you can see on the left-hand side of the slide, that all 4 of the genes contribute to the PA phosphatase activity in the cell. But the experiments shown on the right-hand side of the slide show that only the PA1 PA phosphatase contributes to the biosynthesis of triacylglycerol. So in this slide, I show the cellular locations and roles of the 4 PAP enzymes. So as I just mentioned, PA1 is a peripheral membrane protein that's involved in lipid synthesis. The APP1 PA phosphatase is associated with actin patches, and it's involved in endocytosis. DPP1 and LPP1 are integral membrane proteins that associate with the vacuole and Golgi, respectively, and they're involved in lipid signaling. So our work has focused in the last 13 years or so on the PA1 PA phosphatase because of its impact on triacylglycerol biosynthesis. So this slide shows the relationship of the PA phosphatase reaction with respect to overall lipid metabolism. So in growing yeast or in your early career scientists, when you're actively growing, the phosphatidic acid is primarily shuttled to CDP diacylglycerol for membrane phospholipid biosynthesis. But in my phase of growth, like in yeast, as we get older, the PA is primarily shuttled to diacylglycerol for the production of storage lipid, triacylglycerol, right? And the triacylglycerol is stored in lipid droplets. I'd like to point out that the substrate PA and the product diacylglycerol has a function in membrane structure. Right? And they also have bioactive functions. For example, diacylglycerol is an activator of protein kinase C. And then a major role of PA is in the transcriptional regulation. So I'm going to come back again to that later. So what happens when cells don't have PA1 PA phosphatase activity? Cells are very, very sick. Right? As you'd expect, they have high levels of PA because they're not being converted to diacylglycerol, right? And so they have low levels of triacylglycerol and they have defects in the formation of lipid droplets. The high levels of PA actually cause an increase in the expression of phospholipid synthesis enzymes, and the cells make more membrane phospholipids. And if you look at the nuclear membrane, it expands. But there's many other phenotypes that you can see on the slide here. As I mentioned, the cells are very sick, and so ultimately they have an apoptotic phenotype, and they have a decrease in their lifespan. So what about PA phosphatase in mice and humans? Right? What you see here are a bunch of the phenotypes. For example, mice have lipodystrophy, peripheral neuropathy, rhabdomyolysis, which is also shown in human. There's a lipin 2 isoform that defects result in anemia and type 2 diabetes. I showed the photograph of Karen Rui on the top left-hand side of the slide. She was responsible for the identification of the lipin-1 gene and the lipin-1 protein. But of course, at that time, the molecular function was not known. But she recognized the homology between mouse and human and yeast, and many of the things that she recognized in her seminal paper was very useful to our research. So on this slide, I have an alpha-fold structure of the PA1-PA phosphatase. And you can see the squiggly stuff in the structure, and the squiggly stuff means that there's about half of the protein is unfolded. And the intrinsically disordered regions of the protein have precluded the determination of a bona fide structure for the protein. But luckily, Michael Arola's group determined a structure for a PA2 homologue that's found in tetrahymena, right? And it doesn't contain the intrinsically disordered regions which allowed his laboratory to determine a structure. And of course, the alpha-fold structure that you see, it must be based upon the real structure. Right? So there are other features in the protein that I will talk about. And for example, there's of course, there's an N-lip domain and an HAD-like domain that form the catalytic core, which was shown in the roller structure. There's also an amphipathic helix that's involved in membrane association. There's an acidic tail, AT, that's involved in the interaction of the NEM1 SPO7 complex. There's an essential tryptophan that's required for the function. And there's an RP domain that's involved in the phosphorylation of the enzyme. An undergraduate student named Parth Sharma, along with my graduate student Jordan Stuckey, examined the alpha-fold structures of many PAP enzymes, as well as many non-PAP enzymes that have the haloacid dehalogenase domain. And what you see on the slide here is that the active site of these HAD-like enzymes contain 4 motifs. So there's 3 motifs in addition to the conical DXT motif. I'd also like to show you that all the PAP enzymes have an N-lip domain that's required for activity. But non-PAP enzymes do not have the N-lip domain. So there's something very special about the N-lip domain. And I will tell you that the PAP enzymes work on their substrate, PA, which is part of a membrane, but the non-PAP enzymes work on substrates that are water-soluble. So there's some indication there. So Jordan Stuckey did a number of studies where he made mutations in the 4 motifs. And the experiment shown on the top right-hand slide is showing you that mutations that he made, he expressed and purified the protein. And you can see that if you make a mutation in any of the motifs, that you lose activity. He did some in vivo studies that are shown on the bottom of the slide. And he showed that if you express the mutant enzymes in a PA1 mutant that has various phenotypes, for example, those mutants cannot correct the defect in triacylglycerol biosynthesis, and they cannot correct the temperature sensitivity defect of the PA1 mutant. So both in vitro and in vivo studies, confirm the importance of the active site motifs. Gil-Soo Han has been very interested in looking at the role of the NLIP domain and its interaction with the HAD-like domain. And he asked the question, could you possibly express, constructs where you only had one domain? And if you expressed constructs that had, each of the domain, could they get together in vivo? And he scored the ability of them getting together or clicking together by looking at the temperature-sensitive phenotype. And so on the right-hand side of the slide, you can see where he expressed each construct, individually. You could not correct the phenotype at 37 degrees. But when you do the split expression, they get together. So his current studies are looking at the residues that are involved in the interaction. Another student in my laboratory had done an analysis by cutting up the protein and also by using bioinformatics. And this is before the AlphaFold structure came out. And Young-Hee Park discovered a WW domain in the protein. And she discovered that there's an essential tryptophan that I showed you before that's essential for the function of the enzyme. But it's not involved in the activity. But it is required for in vivo function. Now, a great part of our research deals with how is this enzyme regulated? If we could understand what makes this enzyme tick, maybe we can, cure some of those lipid-related diseases. So location, location, location is the key thing that regulates PA phosphatase. Right? And so the enzyme, when it's first made, is unstable, but nature has come up a way of stabilizing the protein by phosphorylating it. However, the phosphorylation sequesters the enzyme in the cytoplasm away from its substrate, phosphatidic acid, that's at the ER membrane. So in order for the enzyme to be active, it must be recruited and dephosphorylated to the ER membrane, and that's done so by the NEM1 SPO7 phosphatase complex that Simeon Sinison identified in 1998 before it was known that the substrate was PA phosphatase. So after rounds of dephosphorylation, the enzyme is unstable and it's degraded by the 20S proteasome. So phosphorylation is really critical to the enzyme function, because not only does it stabilize the protein, but the phosphorylation itself will allow the enzyme to be recruited to the right membrane, right? And so the phosphorylation is a place of identification because it's a substrate for the complex that's found at the ER. So what you see on the left-hand slide, you can see the intrinsically disordered regions that are highly decorated there with green spots. And those are the sites of phosphorylation. On the right-hand side of the slide, you see that there are 66 phosphorylation sites in the protein, right? And the symbols next to each site indicates the protein kinase that we've identified to phosphorylate that site. So you can see there's a lot more work to uncover the other protein kinases. You'll also see, for example, with serine 602, there are 4 protein kinases that phosphorylate that site. So nature's come up with a way to make sure that site gets phosphorylated during different phases of growth or different growth conditions, because it's critical. Okay? So the phosphorylation is highly complex. Right? There's work by hierarchy phosphorylation, and this enzyme is subject to this type of regulation. So the phosphorylation of one site can either stimulate or inhibit the phosphorylation of another site. So it's very complicated, a really crazy situation. On top of that, the NEM1 SPO7 complex is also phosphorylated. So let's talk a little bit more about some of the work that Jordan Stuckey, he identified this RP domain, and he found that if you delete the RP domain, there was greater PA phosphatase activity. And why was that? So previous work from the lab found that the phosphorylation itself inhibits activity. And so by removing this domain, he found that there was an alleviation of this inhibition. And a mass spec analysis shows that there's about a 50% reduction in the overall phosphorylation state of the protein. In the experiment on the right-hand side of the slide, Jordan showed an in vitro experiment that if the enzyme is lacking this RP domain, that the phosphorylation by Pho85, Pho80, which is a major protein kinase, right, is reduced significantly. So the RP domain is very important. So let's talk about the NEM1 SPO7 complex that was discovered by Simeon, my collaborator. Okay? And so the alpha-fold structure of the complex is shown on the left-hand side. NEM1 also happens to have an HAD-like domain. And recent studies in my lab by Emily Bostrom found that it also has the 4 motifs that Jordan found in PA1. Please visit Emily's poster, number 278, on Tuesday, Monday, and you can hear her talk. So other studies in my lab identified the residues that are important for the interaction of NEM1 and SPO7, and the residues that are critical for the interaction of SPO7 with PA1. Any mutations in these interaction sites will give rise to mutations just like the ones in PA1. So in other words, the NEM1 SPO7 complex is sort of a master regulator of PA1 PA phosphatase activity. So that's critical. Let's get back to Susan Henry. So the studies of Susan Henry during the 1980s, that her laboratory discovered some very important aspects of the transcriptional regulation of lipid metabolism. And she found that many phospholipid synthesis genes contain a UAS-ENO element in their promoter. And it's the place where there's an ENO2, ENO4 complex that binds to the promoter and drives transcription. They also discovered a repressor called OP1. And if the OP1 repressor interacts with ENO2, transcription is attenuated. So what's the connection with PA phosphatase? So it turned out that OP1 function is also regulated by location. And so when PA levels are high, OP1 associates with a protein called SCS2 at the ER membrane. Right? And if PA levels are low, for example, when you have PA phosphatase activity, then OP1 can translocate, bind to ENO2, and attenuate transcription. So the whole pathway that we worked on in the 1980s comes full circle. There was an experiment that I'm just going to briefly mention here by another undergraduate in my lab, Andrew Privalov. And he has a poster tomorrow, number 212. And he looked at the expression of CoA1, which encodes phosphatidylserine synthase. So for example, if you look on the Western blot at the top, the vector control is a PA1 mutant. So in a PA1 mutant, you would have very high levels of PA, right? And the high levels would bind OP1. And that allows for transcription of all those UAS-containing genes. So here you see there's a big blob on the Western blot for CoA1 expression. Now, of course, in the wild-type condition, you see a lower level of expression, and that's because of the regulation. So he also did experiments with phosphorylation site mutations, and you can talk to Andrew about his experiments. So for the remaining few minutes, I want to talk about some additional studies by Jordan. So there was work by a number of groups that were looking at the reutilization of FDA-approved drugs. And it was found that sertraline, which is also referred to as Zoloft, is a serotonin reuptake inhibitor, and it inhibits the growth of Saccharomyces cerevisiae as well as other pathogenic fungi. And some work by Matthew Sachs showed that when you feed these cells with sertraline, they have defects of membrane expansion, they have defects in lipid droplet formation, and these were the phenotypes that I talked to you about that the PA1 mutant shows. Jordan said to me one day, boy, these are phenotypes that we see in PA1. Do you think it's possible that sertraline's an inhibitor of PA phosphatase? And I said, Jordan, go for it. So to make a long story short, and I put Jordan through hell, and I made him do enzyme kinetics, and you can see by the experiments on the slide here that sertraline's a non-competitive inhibitor of PA phosphatase activity. He did molecular docking experiments, and he showed, at least through bioinformatics, that it associates with the HAD like domain, but not at the active site, consistent with the non-competitive inhibition. I said to Jordan, you have to prove that it happens in vivo. And so we said, okay, Dr. Carman, I'll express the PA1 gene, in the cells, and we can alleviate the sertraline-mediated inhibition of growth. And that's what's shown in this slide. So what about all these experiments we do? We do structure-function studies. Why? Because we hope that the information can be useful someday for chemists to come up with drugs, can either activate or inhibit the activity. Through these studies, we found that, the regulation of the yeast, PA phosphatase, and the human are not identical. There are differences, but these differences can be used to our advantage. And then, could sertraline or other FDA-approved drugs be used as an activator or inhibitor? So these are things that we're looking forward in the future. So again, let me thank the Carman clan who did all the work. I want to show you Jordan Stuckey's picture is at the top there in the middle. That's him. Right? On his right is Joe Stuckey. On his left is Ginny Stuckey. They were postdocs in my lab. And after they left my lab and they went to Michigan, they had Jordan. And I'm so grateful that Jordan came to my lab. I want to have a special thanks to Gil Suhan, who has been a driving force in my lab. And I want to leave you with Carman mottos. Just because it's published doesn't mean it's fact. Don't assume anything. Most importantly, you don't detract from your own accomplishments by acknowledging those of others. I can't pay back Bill Dohan, but I can pay it forward. I can pay it forward to the next generation. Thank you.